These are the lecture notes for Chapter 20 on viruses, bacteria, and archaea. There are 5 sections. So the first section is on viruses and a couple of other disease-causing substances. They're really not living things, but viroids and prions. And then we're going to talk about emerging diseases, including SARS-CoV-2, which is the cause of COVID-19. Then we are going to focus on prokaryotes. So that's going to include the 2 domains of living things called bacteria and archaea. So the sections 3, 4, and 5 actually deal with living things, which are made of cells, whereas viruses, viroids, and prions are not technically living things. So we'll start with section 1, and we will begin with viruses. For some reason, I'm trying to do the highlighter, sorry. All right, well, I don't know why it's not letting me, but that's okay. I can point. So just make sure for if you're taking notes, and I do encourage you to take notes. You don't have to copy everything, but typically what I do when I'm recording is I highlight what you should write, but I guess I can just point and tell you. Make sure that you know that viruses cause disease, but they are not considered living things because they need a host cell in order to reproduce. They can't reproduce without their host cell. They also are not cellular. They're non-cellular. But like living things, they may have either DNA or RNA as their genetic material or their genome. And then there's just a little note about the word virus was used the first time by Louis Pasteur, who you may have heard of developing the process of pasteurization that we use for canned foods. And he also discovered transformation, which we'll talk about in this same chapter. So remember Louis Pasteur, because we will be talking about him a little bit more. But he used the word virus for the first time, and that word is Latin for poison. So some of the diseases in humans that are caused by viruses rather than bacteria or fungi or protozoans are going to be STDs like AIDS, which is caused by human immunodeficiency virus, genital warts, which is caused by HPV, human papillomavirus, genital herpes, the Zika virus causes Zika disease. There are childhood diseases which we get vaccines for, and we also get we can get a vaccine for herpes and genital warts. But mumps, measles, chickenpox, which can lead to shingles later in life, so you can get vaccines for chickenpox, German measles, and then there are some respiratory diseases which are caused by viruses that you cannot be vaccinated against, and that would be the common cold and the flu or influenza. And then there are SARS viral diseases, which stands for severe acute respiratory syndrome. And the one that's caused by the virus SARS-CoV or COVID-2 is the one that causes COVID-19. Okay, then there are skin diseases that are viral, like warts, fever blisters, and shingles, which is caused by the same virus that causes the chickenpox, which is called varicella zoster. There's gastroenteritis, which just mainly means inflammation of the GI tract. And that can be caused by a virus, but it can also be caused by bacteria or a protozoan or even a fungi. And diarrhea, same thing. So these two digestive conditions can be caused by viruses as well as other pathogens. Nervous system diseases that are caused by viruses: poliomyelitis, rabies, encephalitis, Zika, and there is smallpox, there is hepatitis, some types of cancer, mono or mononucleosis, yellow fever, dengue fever, conjunctivitis or pink eye, hepatitis C, and Ebola. So if they're caused by a virus, you can't treat them with antibiotics, but you can sometimes get a vaccine that will prevent you from getting the virus. All right, so for viral structure, every virus has at least two parts. At least two parts. Some viruses have more, but there's at least a core, and that core contains DNA or RNA, and then a capsid outside the core. The capsid is made of protein. So there's always a nucleic acid core, and we can classify viruses based on whether or not it's DNA or RNA, but also whether it's single-stranded or double-stranded, because viruses are a little different from what we learned in Bio 111. We learned that RNA is always single-stranded and DNA is always double-stranded. But in viruses can have single-stranded DNA and double-stranded RNA. And then the capsid is composed of protein, and then sometimes there's an envelope composed of a plasma membrane, like a phospholipid bilayer. And that envelope, some viruses have it and some do not. So looking at this chart, if you were to see a particular virus particle under an electron microscope, because they're too small to see with a light microscope, if you look at the outer covering of the virus, it's always going to have a protein capsid. Sometimes it will have an envelope of plasma, similar to a plasma membrane. And then for its inner core, it's always going to have a nucleic acid, either DNA or RNA, either single-stranded or double-stranded. And sometimes it will have enzymes, various proteins or enzymes like, reverse transcriptase is one that you have in some viruses, but DNA polymerase, RNA polymerase, any of those enzymes that are used in converting DNA into a protein. Here's some images with electron microscopes, and then below you see illustrations that are labeled of some typical viruses. There's an adenovirus here on the left, and you can see the capsid is kind of has a polyhedral shape or a geometric shape to it, and then it has DNA on the inside, a DNA core. And then you have a bacteriophage. The bacteriophage is a special name for a type of virus that only infects bacteria. And this bacteriophage has a protein capsid. This whole green helical shape here, and then as well as the tail and the legs, the tail fibers, they look like legs to me. All of that is the capsid, and then it has DNA on the inside. And on the next slide, we have, this will be slide 8, we have an electron microscope image of tobacco mosaic virus, which doesn't just infect tobacco, it can infect many types of plants, tomato plants, orchids. There's a lot of different plants that it can infect. But it's an RNA virus, and it has a helical capsid. And then we have the influenza virus, which is an animal virus. It can be other animals, but definitely it's a human virus. And a lot of animal and human viruses will have, outside the capsid, they will have an envelope. So you can see an envelope here. You see the capsid, which is green, and then you can see the RNA inside the capsid. All right, so viruses are, and you would need to put this in your notes, they are obligate intracellular parasites. Obligate intracellular parasites. I do not understand why I can't. I'm irritated about that. Okay, we're going to try this again. Yay! Okay, so now I can highlight. Make sure in your notes that you know that a virus is an obligate intracellular, meaning it lives inside of cells, and it's a parasite. So obligate means it must live inside of a cell, and you can't really say live because a virus isn't a living thing, but obligate intracellular parasite, that's what we call viruses, and because they cannot reproduce outside a living cell. And the only way you can culture a virus is inside living cells. So for animal viruses, a lot of times we'll use a chicken egg to culture or grow viruses, or tissue culture in a petri dish or test tube. All right, life cycle. Even though they're not true living things, they do have a life cycle or reproductive cycle. Basically, a virus has to attach to the host cell, and it attaches based on the virus will have surface molecules that attach to receptors on the host cell. And the surface molecules are typically glycoprotein spikes, but there has to be a match between the virus surface molecules and the host cell receptors. That's the attachment phase. Then there's the penetration. Either the virus will inject its DNA or RNA into the host cell, or the host cell will engulf the entire virus by endocytosis. So that's penetration. Then there is biosynthesis, where the host cell will synthesize the new viral components, the nucleic acid and the protein capsid, and any of the other components of that virus. The maturation stage is when those components are assembled, and release is when the new viruses that have been produced exit the host cell, either by lysis, which means that they basically kill the host cell, or by budding, and that's a type of like exocytosis. They form a little bud off of the plasma membrane of the host cell, and so just a portion of the plasma membrane would be affected. So sometimes when viruses, during that release stage, the viruses will destroy the host cell, and sometimes they won't. Now, we have used bacteriophages in biology, in research, to understand viral life cycles. And we've learned that bacteriophages go through two different life cycles, the lytic cycle and the lysogenic cycle. So during the lytic cycle, the virus reproduces using the host cell machinery, and then the host cell undergoes lysis. The host cell dies, and virus particles are released so they can infect new host cells. But during the lysogenic cycle, the nucleic acid of the virus is integrated into the host genome, and it goes through a period called latency. It goes dormant for a time. And so it can then become active again. The latent viral DNA is called a prophage, and then they have the example here, diphtheria is caused by a prophage-carrying bacterium. So the bacterium has actually been infected by a bacteriophage virus, and it produces a toxin that can damage the lining of the upper respiratory tract and cause breathing problems. Here's a picture of those stages. The attachment stage, you can see the bacteriophage has attached to the bacterium, which looks like an E. coli based on the shape. And then the penetration stage, the bacteriophage injects its DNA into the host cell. The biosynthesis stage, the host cell produces viral DNA and viral capsids. And the maturation stage, the viral components are assembled and new viruses are produced. And then the release of those viruses by lysis, and that host cell is destroyed. And then if we look at the lysogenic cycle, if a virus enters the lysogenic cycle, it occurs after the penetration step, okay, in the lytic cycle. So once the viral DNA is injected into the host cell, then is integrated into the host DNA. And we call that now, that viral portion, we call a prophage. Now, that period of dormancy can last for various amounts of time, but sometimes at some point in time, it could be stress, it can be something in the environment, but something will trigger that dormant or latent virus to become active again, and it will become active and enter back into the lytic cycle at the biosynthesis stage of the lytic cycle. And this is slide number 19, and we'll begin with slide 19 in the next recording.